I would like to welcome our distinguished speaker for today, Professor Dr. Kikawis Parang, who's a professor at the Department of Biomedical and Pharmaceutical Sciences, School of Pharmacy at Chapman University, California, USA. We are very pleased to have you here, and thank you very much, sir, for taking out the time. I know it's a bit late at your end as well, but thank you so much for taking out the time and delivering this very important webinar today. So we have had quite a few registrations for this as well, and we have had registrations from various OIC member states. So as time progresses, we'll take Distinguished Scholar webinar is on cyclic peptide basic format of the webinar would be that you would conduct the lecture first, and at the end, we can take a few questions from the participants as well. And we will also be recording this lecture. So for anyone that starts a bit late, they can catch up on it through our YouTube channel as well. So over to you, sir. Thank you very much once again.
Okay, good morning, everyone, and thank you for invitation. I really appreciate taking your time to come to my talk. I'm going to talk about some of the research I've been doing for last few years regarding the peptide drug conjugate to overcome the drug resistance and also anti-cancer chemotherapy toxicity. So I'm from Chapman University School of Pharmacy. It is located in Southern California. And in terms of my talk, I'm going to give you some introduction and then some background information about my research, and then go to the main area, that peptide drug conjugate. So as you are aware, the cancer chemotherapy is one of the main tools that for treatment of cancer, and we all know that we have about 10 million deaths from cancer every year. If we consider COVID-19 during the 4 or 5 years, we have only 6 to 7 million deaths, but for cancer, we have almost 10 million deaths every year. And some of the main cancer are breast cancer and lung cancer that are dominating most of the mortality. Unfortunately, many of the cancer chemotherapeutic agents have a problem in terms of non-specificity and toxicity. And many anti-cancer drugs develop resistance, and we have undesired pharmacokinetic property. And if we look at the travel from the IV injection of anti-cancer drugs all the way from the blood circulation to the tumor, there are many challenges and barriers that anti-cancer drug have to go through. And then, unfortunately, during that process, there are also side effects and problem with the vascular endothelium permeability, cell wall permeability. And so the anti-cancer drug may penetrate to other tissue. And by the time they get to the cancer cell, we also want to make sure they reach to the right target. A lot of anti-cancer drugs have to go to the nucleus of the cancer cells because the DNA replication happening over there. So we see that some of the anti-cancer drugs never reach to the nucleus. And if we look at several cancer targeting tools that we have and have been used, we have liposome, nanoparticle, red blood cells, antibody, multivalent inhibitor, and also peptide. Each one of these have different advantages and disadvantages, and Some of them are complementary to each other. But if we want to summarize in terms of liposome, as you know, they are spherical nanoparticles that can encapsulate hydrophilic drug and also hydrophobic drug. The problem with the liposome is that they have very minimal stability, and we will have leakage of the drugs in blood circulation. There are also polymers like polylactic acid, glycolic acid that are used for the delivery, and they are commonly used. The problem with the polylactic acid and glycolic acid, PLGA, is that they degrade to lactic acid and glycolic acid, and so they have limited loading capacity for the anti-cancer drug. Solid lipid nanoparticles are much more stable compared to liposome and PLGA, but then on the other hand, they have a very limiting loading capacity. But they are used for the vaccine, like mRNA vaccine and other type of smaller, small loading. And my area of this research is cell-penetrating peptides. Cell-penetrating peptides are another approach that these are a small peptides that compose of different amino acids, and they have usually high internalization. They can pass through the cell membrane quickly, and they are biodegradable because they are formed from composed of natural amino acid, and they can be easily synthesized. And the whole concept started from the TAT peptide, that is a protein that is involved in the HIV, and they realized that this TAT protein is able to attack T lymphocyte, and this TAT protein have multiple positively charged amino acid. And then based on that, several other cell-penetrating peptides have been synthesized that compose of hydrophilic amino acid. Most of them are cationic and or hydrophobic amino acid like tryptophan. Many of these linear cell-penetrating peptides, unfortunately, are not very specific, and they are very toxic, and they have a very short half-life because are linear, so they are natural amino acids, so they can be degraded quickly by the cell proteases. So my research has been to develop some cell-penetrating peptides that, first of all, are much more stable and are rigid, so they can survive the degradation enzyme in the body. And then also they have hydrophilic and hydrophobic amino acid at the same time. And the hydrophilic and hydrophobic amino acid that we selected are tryptophan and arginine. Arginine is a positively charged amino acid, and tryptophan is the most hydrophobic amino acid in our body. So we designed many different cell-penetrating peptides that have, for example, hydrophobic head and hydrophilic tail, or hydrophilic head, hydrophobic tail, or there are bicyclic peptides that have one cyclic portion is hydrophilic, one cyclic peptide is hydrophobic, and or we have a head and two tails that is hydrophilic and hydrophobic, or we have alternate hydrophobic and hydrophilic amino acid, or all the hydrophobic amino acid are in one side and all the hydrophobic on the other side. So each one of these design have their own story, so I'm not going to go through each application. But what I'm trying to say that cyclic cell-penetrating peptides have major advantages compared to linear peptide because they are more stable toward proteases, and they increase the cell permeability compared to the linear peptide. Many of cell-penetrating peptide, when they want to pass the cell membrane, they go to the organelle, we call it endosome, and we have endosomal uptake, and this endosomal uptake encapsulate the cell-penetrating peptide, and sometimes cell-penetrating peptide end up remaining in the endosome and they cannot escape. But we realized that several cyclic cell-penetrating peptide we have, they don't have endosomal uptake, or if they have endosomal uptake, they can easily pass the endosome and go to the cytosol and eventually reach to the nucleus. So one group of the cyclic peptide I want to talk about is cyclic peptide that have alternate hydrophobic and hydrophilic amino acid. When I'm talking about W, W means tryptophan, and when I'm talking about arginine, arginine is represented by R group. So we have different number of amino acid, 4, 5, 6, 7, 8 tryptophan and arginine at the same time. And if we have an alternate hydrophobic and hydrophilic amino acid like this, we realize this peptide actually have a cell-penetrating property and they can be used for molecular transporter. On the other hand, if we have all the hydrophilic amino acid on one side and subsequent hydrophobic amino acid on the other side, then we have antibacterial peptide. So this antibacterial peptide have different property because all they have a lot of amphiphilic property. They are not like alternate drug delivery agent. So based on this design, we came up with using alternate hydrophilic and hydrophobic cyclic peptide for delivery of different anti-cancer drugs. And one of these peptides that I'm showing here that has alternate tryptophan arginine has been used for multiple applications. For example, for molecular transporter property, for doxorubicin delivery that I'm going to talk about today. They also have protein tyrosine kinase inhibitors, so they have their own intrinsic anti-cancer activity. They can also be used for delivery of other anti-cancer drugs or other drugs like Taxol, camptothecin, curcumin. We have used also this for delivery of nucleic acid, CRISPR, and negatively charged cell-impermeable compound like phosphopeptide. And they can also be used for designing a nanoparticle. And also this cyclic peptide have this property. They can be used as a stabilizing agent and making sure the protein are more stable in the presence. So how this cyclic peptide really work? So we realized that at higher concentration, this cyclic peptide generate nanoparticle and they self-assemble with each other. And you can see a smaller particle self-assemble with each other and generate larger particle, and this assembly continue and is in time dependent and concentration dependent. So our hypothesis is that this, when you have a cyclic peptide that have hydrophilic and hydrophobic amino acid, this hydrophobic residue will love to be next to each other and generate the self-assembled structure. And this hydrophilic residue like arginine, they want to be exposed to the water. And then if you have a hydrophobic drug, you can encapsulate inside the hydrophobic residue and deliver them to the cell. So this is like a story that Troy horse, that soldiers can go inside the horse and then they can pass through the castle and then release inside the castle, so, and fight with the enemy. So that is our hypothesis that we wanted to test. So in order to test our hypothesis, we wanted to see how we can prove that. And so we use a mother compound. Lamivudine is an anti-HIV drug. And we use a linker and attach it to the fluorescein residue. So we have a fluorescence labeled drug. And then we use several different type of cyclic peptide. And this cyclic peptide, they have hydrophobic and hydrophilic residue like tryptophan, arginine, phenylalanine, lysine. We also use other type of amino acid like negatively charged amino acid as a control. We also have negatively charged amino acid and lysine. So we use different type of combination of amino acid and to see which one of them can be used as a molecular transporter. And as you can see, when we have tryptophan and arginine, 3, 4, and 5, and we have this model fluorescence labeled compound. And we do the flow cytometry, we can see the uptake of this fluorescence labeled drug increase in the presence of tryptophan and arginine-based peptide. Other peptides did not improve or did not significantly improve the uptake. So only this tryptophan and arginine-based peptide, cyclic peptide, were able to deliver our model compound. And when we do the fluorescence microscopy, if you have only the drug, we have a very limited uptake because the drug is very polar. And in the presence of WR4 and WR5 that are alternate tryptophan and arginine, we get a lot of uptake. We can easily see green fluorescence uptake, for example, in leukemia cells. So it shows that the peptide is facilitating the uptake of the cargo molecule that is a fluorescence labeled and can deliver it inside the cell. And if we also look at other model compounds, for example, we use a fluorescence labeled phosphopeptide. Fluorescence labeled phosphopeptide, they have negatively charged phosphate, and then cell membrane also has a phospholipid that's negatively charged. So they have a very limited uptake inside the cell. And as you can see, if we use DAPI, DAPI is a stain that we can stain the nucleus of the cells. And Texas red is showing the cytoskeleton of the cells, mostly cell membrane. And as you can see, we don't have much uptake inside the cell if we use only fluorescence labeled phosphopeptide. If we use a cyclic peptide, you see significantly improved uptake, and you can see green fluorescence inside the cell. In this case, because fluorescence labeled phosphopeptide, they localize mostly in the cytosol. And if we look at the three-dimensional structure of fluorescence labeled phosphopeptide in the presence of the cell, we don't see much uptake. But when we have the cyclic peptide, as you can see, all the green fluorescence can be seen around the nucleus and in the cytosol of the cell, and we have a very good uptake in the presence of cyclic peptide. So how about the cytotoxicity of this compound? If you look at the cyclic peptide, we tested them in different cancer cells, leukemia, colon cancer, breast cancer, ovarian cancer, and up to 100 micromolar, we have a very good viability. So we don't see much cytotoxicity. And we usually use this peptide at low micromolar concentration, and they have a very fast uptake. So we don't usually use 100 micromolar. We usually use less than 10 micromolar. So now I showed you that this peptide is able to deliver a cargo molecule inside the cell. But where this peptide itself end up? So we wanted to track whether this peptide is actually a cell-penetrating peptide. So in order to understand this, we use a linker and attach the fluorescein to our peptide. And we incubate different cells like ovarian cell, cancer cell, colon cancer cell, and breast cancer cell. And we incubate the cells with this fluorescence-labeled peptide. And as you can see, most of the peptide is immediately after 1 to 5 minutes goes inside the cell. And if we look at the nucleus with the DAPI, and then we merge this picture, we see most of the peptide is actually inside the nucleus. So we will have a very high uptake inside the nucleus. So we determined that this peptide actually are nucleotargeting peptide. So we came up with this idea. So if it is nucleotargeting, maybe we can use them for delivery of anti-cancer drugs that target the DNA. So based on what I told you, so far we have cyclic peptide that contain tryptophan and arginine. They can generate a self-assembled nanostructure, and they can encapsulate a small hydrophobic molecule, and they can be used as molecular transporter. So we're looking at the application of this. We were able to deliver negatively charged phosphopeptide and a small molecule by encapsulation. So then we started going to more advanced study, thinking about delivery of anti-cancer drugs. One of the gold standard in cancer chemotherapy is using doxorubicin. Doxorubicin, first of all, is very effective against many different cancers. So it's an anthracycline. It has 4 cyclic group and intercalate to the DNA, and it also has topoisomerase inhibitory activity. Topoisomerase is an enzyme that when the DNAs are super coiled, they mm, break down the DNA and they relax them and then reseal them. So they can, the DNA can undergo replication. So doxorubicin inhibit the topoisomerase 2. So when the DNA is nicked and cleaved, it blocks the resealing of DNA, so the cancer cell cannot have a DNA that is functional, and then we have apoptosis in cancer cell. So doxorubicin bound to DNA and topoisomerase at the same time. And one other advantage is it has a red fluorescence, so it can easily be tracked. I don't need to conjugate it to any fluorescence compound, so I can do all the cell-based assay with the intact drug. The major problem with the doxorubicin is 2 major problems. One major problem is cardiotoxicity. So a lot of oncologists avoid using doxorubicin, especially for people who have cardiovascular diseases. And the toxicity against heart is kind of irreversible and accumulating over time. So if people have cardiovascular disease and cancer at the same time, oncologists try to not to use it. And on the other hand, if they use it for other patients, it also causes cardiotoxicity. And the problem also is there are a lot of doxorubicin resistant cancer cells. So cancer cells that pump out doxorubicin outside the cancer cell. So there is a efflux pump that eliminates doxorubicin from cancer cells. So if we look at the doxorubicin, as I mentioned, is bound to the topoisomerase 2 and generate nick DNA and double strand break, and then we have anti-cancer activity. But on the other hand, in the heart cell, this anthracycline generate superoxide oxygen, reactive oxygen moiety. And this superoxide oxygen eventually converted to hydrogen peroxide and hydroxy radical. and this hydroxy radical undergoes some reaction, we call it Fenton reaction, that cause toxicity in the heart cell. So how we can use some of our peptide to minimize the cardiotoxicity of doxorubicin. On the other hand, also we can generate compounds that are active also against doxorubicin resistance cell. So during the last few years, we designed a lot of cyclic peptide and cyclic linear peptide that have hydrophobic and hydrophilic amino acid and attached to different type of linker to doxorubicin. And I'm not going to go through each one of these. I give you a couple of examples that and show you the activity in terms of cardiotoxicity, antiproliferative activity, and activity against doxorubicin resistance cell. So I'm going to talk about cyclic peptides that have alternate hydrophobic and hydrophilic residue attached to doxorubicin, and also some cyclic peptides that have hydrophobic residue and hydrophilic residue and attached to another linear peptide. I call this hybrid cyclic linear peptide, and then is attached to the doxorubicin. So the simplest model we designed is the cyclic peptide I showed you at the beginning. This is a WR peptide and attached with a linker to a doxorubicin. And then when we incubate this doxorubicin peptide conjugate with different cell lines, like for example, ovarian cancer cell, SKOV. If we use only doxorubicin, as you can see, it has a fluorescence. It goes directly to the nucleus of the ovarian cancer cell. If you have a peptide drug conjugate after 1 hour, we also have high localization inside the nucleus of cancer cell. After 24 hours, most of this doxorubicin is effluxed out of ovarian cancer cell. But after 24 hours, when we have the peptide drug conjugate, you can see high retention of the peptide drug conjugate inside the nucleus. So this shows that the peptide drug conjugate is behaving like doxorubicin. It has high localization in the nucleus of ovarian cancer, but it's not effluxed out of the ovarian cancer cell, and it has a high retention inside the nucleus. We designed another conjugate, a little bit longer peptide, WR9, and attach it to doxorubicin. And if we look at the cellular uptake mechanism, as I mentioned earlier, there are direct penetration, but there are endocytosis pathway. Endocytosis pathway, there are several type of vacuole like endosome creation by clathrin, kevolate, micropinocytosis, so they can encapsulate the nanoparticle and take them inside the cell and then deliver them inside the cell. And as I mentioned, sometimes they end up remaining in endosome and they never be delivered to the nucleus. So they get trapped inside the endosome. Direct penetration, there are different models because of hydrophobic and hydrogen bonding interaction and ionic interaction of the peptide with the complementary residue in phospholipid that has negatively charged and lipid, and they can go inside the cell. Okay, so the preferred uptake is to have a direct penetration because we don't want our particle to end up in endosome and we don't know the fate. So when we check the endocytosis, we wanted to see whether the uptake of this peptide, the peptide drug conjugate, change in the presence of different endocytosis inhibitor. So there are several endocytosis inhibitors, nystatin, chloroquine, chlorpromazine, methyl beta-cyclodextrin. These are inhibiting different pathways of endocytosis like kevolate pathway and or clathrin mediated pathway. So we use all these inhibitors. So we wanted to see if we inhibit the endocytosis, do they impact the uptake? So we saw that there is a little minimal impact in the endocytosis of this peptide. Means that this peptide actually bypass the endosome. And so we have endocytosis um, bypassing and direct penetration. So that's one advantage. And then we wanted to see how this peptide actually getting hydrolyzed. So we have ester moiety and amide moiety, and we look at the human plasma, and we saw that improved release of doxorubicin over time, and then degradation of the peptide. So percentage of remain undegraded slowly decrease. So, and if we look at the HPLC, so if we have a doxorubicin conjugate, 4 hours and free doxorubicin, after 12 hours, we have more free doxorubicin, like less doxorubicin conjugate, and eventually after 48 hours, everything converted to free doxorubicin. So this shows that this conjugate actually is converting to free doxorubicin and acts as a prodrug. So now we have a conjugate that we know that is bypassing the endosome, and we wanted to see how this can be used in vivo in animal study. So for animal study, we wanted to have multiple functional property in our nanoparticle. So first of all, we want to have a targeting moiety. We wanted to target cancer cell. So if we use only peptide drug conjugate, it's likely that it also goes to other organs. So we wanted to attach a targeting moiety like folic acid. Many cancer cells, they have a folic acid receptor and overexpressed compared to the normal cells. So if we attach a folic acid to our conjugate, we will be able to target folic acid receptor. And then on the other hand, we use a polyethylene glycol to improve the circulation time of the peptide and improve the stability. PEG is used for capping of the peptide in several types of vaccine and protein. So we wanted to have a longer circulation time. Also, we wanted to have a fluorescence tag to make sure that we can track it. So we designed this cyclic peptide that have a folic acid that target cancer cell. We have a pegylation that it can increase the circulation time, and then we have a CY55 that is a stain that we use to make sure that we can track and monitor the uptake in the cancer cell. So we use a mouse model that has two types of tumor. One tumor on the left-hand side xenograft mouse model that is HEK293 tumor that doesn't overexpress folic acid. On the right-hand side, it has a tumor that overexpresses folic acid. It's a HeLa tumor. And then when we inject to the animals, and we see that the conjugate is localized inside the tumor that overexpress folic acid. And if we remove different organs, we see most of the localization in the HeLa tumor that overexpress folic acid. We also have some uptake in the kidney and liver, but most of the uptake was inside the tumor. And on the other hand, we wanted to deliver the drug. So in addition to peptide and imaging agent and a stability agent, PEG, and folic acid targeting moiety, we also attached the doxorubicin, the drug, at the same time. And then we injected to the animals, and then we see that within 1 hour, the compound is localized inside the tumor, and then starting eliminating inside the bladder. So 1 hour, a very short period of time, most of the compound localized in the tumor. And if we look at this model, after 1 hour, you can see the tumor, that compound is localized inside the tumor. And then on the other hand, if you look at the 3 hours model, you see that just a moment. Let me share the screen again. I'm sorry for technical problems. So if we look at the animal study again, okay, after 3 hours, as you can see, the compound is starting release inside the bladder. Okay, so the compound is actually is very fast uptake in the tumor, and then slowly released inside the bladder and eliminated from the body. So we wanted to also evaluate the hybrid cyclic peptide that have cyclic structure and linear peptide attached to conjugates. And we also want to look at the toxicity to the heart cell and ovarian cancer cell. And as you can see, the uptake in the ovarian cancer cells is very high, and then we don't have much uptake in the heart cell. And if we look at the antiproliferative activity of this peptide, you look at the doxorubicin and look at the conjugate, we have very comparable activity for the conjugate and doxorubicin. This is both in the leukemia cell and also in the gastric cancer cell. And if we look at the fluorescence microscopy, you see the doxorubicin is uptaken, but in the conjugate, we have a very high uptake at the same time. So, the antiproliferative activity of the conjugate was very similar in cancer cells, and the uptake was much higher. But if you look at the heart cell, H9C2 is that myocardium cell. As you can see, doxorubicin is quite toxic, okay? But then our conjugate showing a high cell viability. All of our conjugate are showing high viability. If you just use a physical mixture of peptide and doxorubicin, they are very similar to doxorubicin. Similarly in kidney cells, so doxorubicin is very toxic, the cell viability decreases, but our conjugate are not very toxic. And if you can see this also confocal microscopy, that you have pre-doxorubicin and then peptide drug conjugate. In the heart cell, we don't have much uptake of peptide drug conjugate, but in the pre-doxorubicin, you have a very high uptake. And if we look at the IC50 value, if we compare the doxorubicin and conjugate, they have a very similar IC50. In the resistant cells, you see the conjugate is very active. Doxorubicin is not active. This is another resistant cell. Doxorubicin is not very active. The conjugate is very active. So it shows that the compound is very active against resistant cells and is not also toxic against heart cells. So if we did also confocal microscopy in the presence of different endocytosis inhibitor, and we see a still uptake. So we have some minimal less uptake compared to when we don't have no inhibitor, but it's not very significant. We still have a lot of direct penetration of the peptide. Finally, we designed another cyclic peptide. This cyclic peptide have actually a different design. So we use the antimicrobial peptide. We have all the positively charged in one side and all the hydrophobic on another side. And we have also a linear antimicrobial peptide, and we combine them. And when we combine them and we test them against different resistant bacteria, it completely lost the antimicrobial activity. But then we realized that this peptide actually is able to deliver different anti-cancer drugs into the cell. So when we use this peptide and use the doxorubicin, the cell viability decreased. Doxorubicin alone did not have very active activity against doxorubicin resistant cell. But in the presence of this peptide, we see the cell viability significantly decrease, showing 20 to 25% reduction. If we go to 72 hours, we get much higher reduction. So we wanted to see how this peptide is uptaken. So we attached the fluorescence labeled peptide, and then we did see the time-dependent and concentration-dependent uptake of this peptide. And as we can increase the time, we see more uptake, as is shown by the fluorescence intensity. And also the concentration increase, we have more uptake. And then finally, we attach this peptide to the doxorubicin and look at the antiproliferative activity against cancer cell, like breast cancer cell, and we compare with doxorubicin. And you can see here the conjugate is even comparable or better activity than doxorubicin in different cancer cell lines. So doxorubicin alone is active, but then for example, in MCF, our peptide is much more active compared to doxorubicin. On the other hand, if we look at the activity against the doxorubicin resistances, so doxorubicin is not active against this resistance cell, but then the conjugate, as you can see, is still very active, especially at longer period of time, like 72 hours. So we can see that the significant enhancement in activity when we have peptide drug conjugate up to 87% compared to 17% doxorubicin. How about heart cell? When we look at the heart cell, doxorubicin is causing a lot of toxicity, but then our conjugate is not showing any cell viability. So no significant cardiotoxicity up to 8 micromolar. So this is exactly what we were looking for. We were looking for a conjugate that doesn't show cardiotoxicity, but is active against doxorubicin resistance. So what's the mechanism? Why it's not active against heart cell? So the heart cell have a receptor, it's a calcium-dependent receptor. We call it ryanodine receptor. So doxorubicin is able to bond to the ryanodine and increase the efflux of the calcium from the receptor. And then we have calcium leak, and the homeostasis of the heart cell will be destroyed, and that's why it's causing toxicity. On the other hand, the conjugate is not able to do the same thing. So doxorubicin, you have a lot of release of calcium, but then our peptide conjugate is like an antagonist bound to the ryanodine receptor, and it doesn't do anything. It just blocks the activity of the receptor. And if we look at the cellular microscopy, doxorubicin doesn't get into the doxorubicin resistant cell. if we have a physical mixture, we have limited uptake, but when we have a conjugate, we have a lot of uptake inside the doxorubicin resistant cell. In the heart cell is reversed. So when we have a heart cell, we have a lot of uptake of doxorubicin and also a physical mixture, but the DOX conjugate doesn't have much uptake inside the nucleus of heart cell. And if we look at the flow cytometry, the same thing. Doxorubicin is not uptaken by the doxorubicin resistant cells, but then on the other hand, heart cell, we see a lot of uptake by doxorubicin, but limited uptake by the conjugate. Conjugate, on the other hand, it has a lot of uptake in the doxorubicin resistance. So we did some apoptosis assays to see what the mechanism. So if you look at the doxorubicin and see which part of apoptosis is impacted. And as you can see, doxorubicin is impacting the early apoptosis, and it doesn't have much impact on late apoptosis. In the terms of conjugate, you see that there are some early apoptosis, and then we have a late apoptosis. If I want to show this in the graphical manner, so if we see the doxorubicin, in the doxorubicin resistant cell, you see that is not impacting apoptosis so much, but the conjugate has a lot of late apoptosis. In the heart cell, on the other hand, doxorubicin is causing a lot of early apoptosis, but the conjugate has no impact on the apoptosis. So this is also confirming our result that the peptide drug conjugate is not causing problem for heart cell, but it's causing problem for doxorubicin resistance cell. And we did also some hydrolysis study in serum and see how the peptide is hydrolyzed to the doxorubicin, and we can track the release of doxorubicin within 48 hours. All the conjugate is hydrolyzed to the doxorubicin. And this hydrolysis is also acidic dependent. So because we know the cancer cells have a lot of metabolism and they have acidic environment. So we can see that in the PBS that has a pH of 6.5, a lot of compound is converted to doxorubicin, but on the other hand, in the physiological pH is much more stable. And we also did the intracellular hydrolysis study to see how the peptide is intracellularly hydrolyzed. And we can see the half-life intracellular is about 24 hours. So the conjugate is slowly hydrolyzed, and we have the formation of doxorubicin. Okay, so I just want to end my talk that we synthesize a lot of cyclic linear peptide and also cyclic peptide that they were not very toxic. They were active against cancer cells, similar to doxorubicin, but they were more potent against doxorubicin resistant cells, and they did not have the dose-dependent cardiotoxicity. And then we were able to generate this prodrug that slowly released and converted to doxorubicin. And so we were able to avoid the efflux mechanism of doxorubicin and also organ-specific cardiotoxicity. So these are some of the people that involved and collaborated in this work, and many other postdoc students, graduate students, Dr. Tiwari, Dr. Mandal, Dr. Lohan, Dr. Muzaffari, Dr. Park, and Riley, and also funding support for different organizations. And we just recently got a new NIH grant to continue this work. So I'm going to stop here, and I'd be happy to answer any question you have there. Thank you so much for your attention.
Thank you very much, sir. So to the participants, if you have any questions, just drop them in the chat box and I'll read them out to Professor. Also, sir, if you have the consent, I'll share you can share the presentation with me as well, because a few of the participants have requested the slides. So,
Yeah, I can send the presentation to you
perfect.
and you can email to everyone who
I will do that.
interested. Yeah. Uh, I'm reading the chat, most of the people asking for a presentation, but then there is a question that we investigate the toxic effect of doxorubicin. We look for method which reduce the toxicity without the reduction of the therapeutic effect. Definitely that's what we're doing, that is our goal. And just to let you know, there are other methods to reduce the toxicity of doxorubicin. For example, there are liposomal doxorubicin available commercially. We call it Doxil. Uh, but then, and it reduced the cardiotoxicity. But as I mentioned, the liposomal doxorubicin is not very stable. They also a formulation of doxorubicin in liposome and pegylated to make it more stable. So there are some advantage to using some of these approaches, but as I told you, we want to also be active against doxorubicin resistant cells. So many of these approaches, they release only doxorubicin and they cannot block the efflux pump. So yeah, I'd be happy to collaborate if you reach out to me by email.
We have another question by Sana Khurshid. What is the optimum number of residues for an effective cyclic peptide?
Yeah, that's a great question. So we have started from 10 amino acid WR5 and we've gone all the way to WR10. Uh, we believe that 10 is enough. If you increase the number of hydrophobic and positively charged amino acid, definitely you have a much higher uptake. But on the other hand, as you increase the number of the positively charged amino acid, there is a chance that you increase the toxicity. So what we found that if we have a very low number of amino acids like WR3, the uptake is minimal. So WR5 was optimal and it was not toxic. But if you go to WR10, you get a very good uptake, but then there is a possibility of toxicity. So I say around 10 amino acid.
We have another question by Ijazul Haq. Can we use other anti-cancer drug other than this doxorubicin to incorporate into this model?
Yes, definitely. So we have tested the camptothecin, we have tested paclitaxel, and we just published curcumin. So if you want to, for example, curcumin has a very low water solubility. So we wanted to see whether we can improve the bioavailability of curcumin. So you can use any different type of anti-cancer drug, especially if you want to target the nucleus of the cell. So if your target is to use some of these anti-cancer drugs, and many of them are like paclitaxel and camptothecin, and they are all mitosis inhibitor. And so we have published those, but there are many other possibilities. We also using epirubicin. Epirubicin is another anthracycline. Another one is mitoxantrone. And so there are many possibilities in that aspect. Yes, good question.
Another one is by Dr. Faisal Gulzar. What parameters should be considered in the selection of conjugate?
Uh, Parameter collection of conjugate, for example, when we use a hybrid cyclic linear peptide, we found out when we have a hybrid cyclic linear peptide, we have a much better activity against doxorubicin resistant cell, and also is not toxic against hard cell. If you only have a cyclic peptide, you get similar impact, but the impact was lower. Another parameter you have to look is in the linker. So if you want to conjugate to be very stable, so you want to use a linker that is much more stable. So we have used ester and amide linker and also disulfide linker. We find out when we use a disulfide linker, we kind of lose the activity. So we move to the ester linker and amide linker. So depend how much stability you want and whether you want to decrease the cardiotoxicity and what type of drug and where do you want to deliver it. So if you want to deliver a drug into the nucleus, definitely this approach is good. But if you want to deliver a drug to the cytosol, some of this approach can be used for DNA delivery, but for if you want to use it for siRNA delivery that is end up to the cytosol, you have to use different type of peptide. So the peptide nature is also important where this conjugate is end up. So all the peptide linker and the drugs, all these parameters that you have to think about. And whether you want a targeting moiety, you want to target the cancer cells, you can add different type of targeting moiety. So I just gave you folic acid, but there are many other different type of targeting moiety like RGD or different type transferrin for different type of cancer. So if you're looking at different type of cancer, you can use different type of targeting moiety.
I think those are all the questions in the chat. So if there are any further questions, they can send it to me over email and I can share them with you as well. So thank you once again, sir, for taking out the time and delivering this informative and brilliant lecture. And inshallah, we look forward to having further such lectures by your good self as a Comstech Distinguished Scholar. So thank you once again, sir, and to the participants that have joined in from various OIC members countries.
Thank you so much. I appreciate that. And I will share the presentation with you. You
Sure.
can share
I'll
with
share it
audience.
with the participants as well. Thank you very much.
Have a good day.
You too.